Good afternoon, everybody. Thank you very much for giving me the opportunity to share with you some experience from other organs than those we are used to bring in connection with COVID-19, but just to bring up how did COVID-19 reach the Institute of Pathology in Basel. So it was in March 2020 when the first patient died at the University Hospital in Basel. And we have been hardly looking for any evidence out of the literature for described cases, for autopsy examinations, and so on and so on. And in spite of the fact that there were already 150,000 recorded deaths in association with COVID-19, there were only 4 case reports of autopsies out of the literature. Then we realized that many medical authorities prohibited even performing autopsies, for example, the Italian colleagues, and at the very beginning also the Germans. Still, performing autopsies is prohibited in Portugal, for example, still now. Since there was no prohibition in Switzerland, and the interest of our clinical colleagues was great, as well as we realized that the patients and their relative consented for autopsies, which is far beyond the usual way it's going in Switzerland. Obviously, there was like a big interest, not only of our clinical colleagues, but also of the affected, to understand what is going on. And after performing the first 20 autopsies together with our colleagues in the Cantonal Institute of Pathology in Basel-Landschaft, even with this primitive method, we already identified the most significant problem with COVID. There were so many findings out of these autopsies suggesting vascular dysfunction. There were many positive effects with this publication, but I think the most important one was that we took away fear from performing these autopsies. And just some weeks later, the Germans allowed to perform autopsies. There was kind of a significant interest of the society towards these issues. And at the end of the day, along with understanding what's going on with COVID-19, this was one of the positive effects of performing these studies. We unlocked the lockdown of science that prevailed at that time. This was also quite contributory, I suppose, to the reviewers of our application towards the call of the Botner Research Foundation, and they granted many of the studies that I'm going to present to you in a second. To make the long story short, so along with this study, we provided tissue to many colleagues throughout the world, Germany, the Netherlands, Italian colleagues, towards colleagues in the United States. And based on those tissue findings, many facts about the pathophysiology of the lung injury in COVID-19 become clear. Along with this, and together with Kirsten Merz in Liestal, we systematically examined the spread of the virus throughout the human body, and actually detected that many various organs are affected by COVID-19, even organs that are deeply buried in the human body, like the bone marrow. There were, for example, here osteoclasts that stained positive for the N antigen of coronavirus. But in this talk, I'm not going to talk on these organs. I'm going to talk on organs and systems that are not listed here: lymph nodes, pancreata, and fatty tissues. So organs that probably were not in the center of the medical attention. Nevertheless, the changes in the lymph nodes were quite interesting for us, because these are mini organs, the sites of active immune response. So this is where the immune system deals with antigens, decides how to react to these antigens, selects proper B and T cells, and actually nothing was known about the lymph node changes in COVID-19. So we systematically collected the lymph nodes of our autopsies and compared the changes there with selected control cases, including cases that histopathologically had reminiscent changes of COVID-19. But collectives were quite similar, with the exception of the quite advanced age of COVID-19 patients, and the COVID-19 patients were more inflammatory. But in terms of other background, the collectives were quite similar, allowing us to draw some conclusions. So we systematically collected these lymph nodes surrounding the bronchi. These lymph nodes are quite brownish. For those of you who are not pathologists, this is because of the filtered dust that comes with inflammatory cells out of the lung to these lymph nodes. So this is basically the dust on the street that we inhale every day, and then it comes here to these lymph nodes and appears brownish. So compared to the usual appearance of a lymph node, the COVID-19 lymph nodes look different. So even for someone who is not pathologist, there's a difference between that image here and that one here. There's considerably more blood here, considerable capillary stasis, and a lot of blood in this lymph nodes of COVID-19. And these funny structures called follicles are actually absent in this lymph node. So this is quite a striking morphologic appearance, and this applies to almost all examined lymph nodes here. So a huge difference between reactive lymph nodes in other conditions and COVID-19. In addition, we were able to see some morphological patterns that are seen in innate immune responses. For example, the huge increase of hemophagocytic histiocytes in COVID-19. So this is like a primitive response towards foreign bodies or towards infected cells. And in addition, we have seen a lot of extrafollicular plasmablasts here at higher magnifications, those large cells. This finding has been also substantiated by an immunohistochemistry, and these plasmablasts either produced IgG or IgM and corresponded to a cellular population that has been described almost 20 years ago in mucosal near lymph nodes that represent a specific way of B-cell response without immunologic memory that occurs very, very quickly, but does not produce very efficient or very specific antibodies, and is called this extrafollicular plasmablastic reaction. We also systematically analyzed the amount of viruses in these collected lymph nodes, and notice that the virus load is significantly lower in the lymph nodes compared to the lungs. So actually approximately 120 times lower in the lymph nodes compared to the lungs. And as shown also by Yasmin, especially in patients that had a longer duration of hospitalization, some of the lymph nodes did even not contain SARS coronavirus RNA anymore. So patients have been able to clear the virus despite succumbing of the disease. To go into deeper detail, we also looked for virus T-cell populations and other populations in the lymph nodes. And one of the most striking differences between COVID-19 and reactive lymph nodes was the dramatic decrease of regulatory T-cells in COVID-19 compared to normal lymph nodes. This is quite important since these regulatory T-cells dampen the immune response and canalize the immune response so that immunity does not harm to the human body. I'm going to extend a little bit on that in a second, because this difference may pave the way towards post-acute sequelae of COVID-19 that are related to autoimmunity and so on. So important finding, and also an important finding here, and I'm going to talk to this also in a second, is that these regulatory T cells do not contain the ACE2 receptor of COVID-19, but they contain an alternative receptor for the SARS coronavirus, namely the neuropilin-1. And this neuropilin-1 plays an important role also in the organs that I'm going to discuss with you later on in this talk. What we also observed was a dramatic increase of hyperinflammatory histiocytes in these lymph nodes, even histiocytes that produce a procoagulative factor, this von Willebrand factor. Also a huge difference between conditions. And also here, even in a lymph node, in an organ that is not supposed to be highly vascularized or be highly related to the circulation, we observed high angiogenic activity in the COVID-19 lymph nodes compared to the controls, which was also an important difference. But the most striking difference, we systematically looked for other viruses in these lymph nodes, was that in a fifth of patients, there was evidence of reactivation of the Epstein-Barr virus within the lymph nodes. And in 1/10 of the patients, there was also evidence for reactivation of herpes simplex viruses in these lymph nodes. And basically, such patterns we have been used to see in severe HIV infections, in AIDS, but we were not aware, and until that time, people were not aware that obviously COVID-19 is a very immunosuppressive disease on itself. And nowadays, there's some evidence suggesting that the reactivation of these viruses, especially the Epstein-Barr virus, may be a wave-paving factor towards long COVID-19. And interestingly enough, antigens of the coronavirus were detected only in a minority of patients, which was also a difference. To go into deeper detail, we compared the gene expression profile of the COVID-19 lymph nodes and the reactive lymph node, showing little overlap. And very strikingly, the most overexpressed genes in these lymph nodes, these are lymph nodes, were genes related to platelet and vascular functions and angiogenesis, and not genes related to immune function. While the genes related especially to T cell functions, they were among the 20 most downregulated genes in this disorder. So obviously, a severe T cell deregulation in COVID-19, which can also be obtained from this schematic overview of the clustering between the controls and the COVID-19, showing actually basically analogous genes. And I would like to draw your attention to one gene here. This gene encodes for prostaglandin D2 synthase. And this gene was relatively downregulated in COVID-19 lymph nodes. And interestingly enough, in mice experiments performed before the pandemics, this gene, downregulation of this gene has been linked to lung injury, vascular permeability, and neutrophilic influx. And what is also known is that the relative downregulation of this gene can be overcome by blocking an upstream enzyme in the synthesis of prostaglandin, namely of the cyclooxygenase 2, which turn out to have some beneficial effects in acute COVID-19 as well. To summarize this part of my talk, we learned lessons from the lymph nodes we examined. There was evidence for a pathological immune response in COVID-19 with extrafollicular plasmablast reaction, suggesting that the immune response was not very efficient, or at least not linked to a immunologic memory. And this is probably one of the explanations, among others, why many people several times developed the COVID-19. So there's not like a fixed immunity in this disorder. There was morphologic evidence for T cell deregulation, especially reactivation of other viruses, and some decrease of the immune dampening or regulatory T cells, evidence of macrophage activation, and we also have seen unexpected gene expression profiles with some potentially interesting targets for treatment. After detecting these things, we asked ourselves, could the lymph node changes in COVID-19 explain somehow also the vaccinational induced lymphadenopathy in COVID-19 with the COVID-19 mRNA vaccine? Just consider that the lymphadenopathy is the most common side effect of the mRNA-based vaccines. The second most common effect is reactivation of herpes zoster infection. So once again, reactivation of latent viruses, but the most common one is the lymphadenopathy. At the beginning of the vaccinational campaign, our medical colleagues were not that used to these side effects. And this is why we have been facing 5 patients in whom lymphadenectomy, so excisional biopsies of the lymph nodes were performed because they were suspected to suffer from lymphomas. Actually, quite significant increase of the left axillary lymph nodes here with splenomegaly, so something that can also be interpreted as a lymphoma. Therefore, we got this tissue, and out of these tissues, we had 3 patients in whom there was sufficient material and consent for scientific research on which we performed this research. Interestingly enough, all these 3 patients were female and had some evidence of immunologic deregulation, as you can see here. So we compared the gene expression profile of those vaccinational-associated lymphadenopathies with lymphadenopathies of COVID-19 and also with reactive conditions, and so that these 3 cases had no overlap with the gene expression profile of COVID-19 itself, but they had a significant overlap with extrafollicular activation of blasts. Even more, performing a CyberSort analysis, we have seen that the vaccinational-associated lymphadenopathies had the highest amount of naive B cells and the lowest amount of memory B cells, which suggests that this COVID-19-related lymphadenopathy may reflect a dysfunctional immune response with extrafollicular activation of B blasts. In order to narrow it down, we looked for the gene expression profiles of these lymphadenopathies compared to COVID-19 and to reactive lymphadenopathies, and discovered many up and down regulated genes. And one of these genes take our attractions. This is the toll-like receptor 10. This is the only known toll-like receptor that is immunosuppressive. And the question is whether the upregulation of this toll-like receptor 10 mechanistically links the lymphadenopathy, of the one hand, to the rare occurrence in switch products. So if a patient developing a significant side effect by a vaccine is vaccinated by another product, usually this lymphadenopathy does not recur. This is the one thing. And the other question is whether the interaction between TLR10 and toll-like receptors that are designed to recognize the spike protein or the single-strand RNA could be an explanation for that. This is something to be done in future, but at least here there's some ideas of a mechanistical explanation. Towards the end of my talk, I would like to touch up on 2 other organs and organ systems that are probably not that in the focus of reports and research, but are a major source of morbidity. This is data from Germany, comparing the incidence of diabetes mellitus type 1 in children before the period of pandemics and in the period of pandemic. So there was an increase of 50% of diabetes mellitus type 1. And this increase was almost doubled when comparing children who had no record of suffering from COVID-19 with those suffering from COVID-19. So 2 million children have been examined. Out of them, 625 developed the diabetes mellitus type 1. And here, almost 50,000 children suffering from COVID-27 developed the diabetes mellitus type 1. So obviously, at least a temporal and maybe causative correlation between those facts. In a study mainly supported by Matthias Matter and by the colleagues from Stanford, we try to address why this may be the case, looking for virus receptors in the beta cells of the pancreas, and actually they were negative for ACAT, but as you may see here, the insulin-producing beta cells of the pancreas also expressed neuropilin type 1. So this is the receptor also on the regulatory T cells and in many other organs. So very important, not expressed by the alpha cells that produce glucagon, but by the beta cells that produce insulin. In an ex vivo experiment, the tropism of the SARS coronavirus type 2 has been shown towards the beta cells here with a confocal microscopy showing that the spike protein is expressed especially on those insulin-producing cells, here presented in a graph. But the most important evidence comes once again from the autopsy material collected in Basel. 4 out of the 7 patients in whom the pancreata were examined for that and were there was sufficient tissue to perform this examination. There was SARS coronavirus mRNA, especially in these insulin-producing cells of the pancreas. So obviously there was virus in the pancreas of those patients. And in an additional experiment, it has been shown that the infection of insulin-producing cells by SARS coronavirus significantly increased their apoptotic activity. So basically those cells die and those cells cannot regenerate once they die, and decrease their activity to secrete insulin. So this part of the research mechanistically explained what has been shown on the epidemiologic part. And this was true also for many other observations in COVID-19, like, for example, the correlation with the obesity and with other risk factors. And now coming to probably the most interesting part of the talk. This is something that was noticed immediately with the beginning of the pandemics. Obesity is the highest risk factor to develop a severe COVID-19 and to be admitted to the intensive care unit or die on COVID-19. This was observed on the populational level, but also in our autopsy study with these 20 patients. It was thought that, fatty tissue may represent a significant reservoir of replicative virus infection because the fat cells express the ACE2 receptor. But actually, and this was true, in a few cases, we were able to detect mRNA of SARS coronavirus within the fatty cells, here, for example, in the epicardium, but this was not the usual thing that we observed. In once again, collaborative study with Stanford that was mainly here led by Matthias Matter from our institute. It has been then shown that the main target of SARS coronavirus infection in the fatty tissue are not the pre-adipocytes and adipocytes, but a special population of inflammatory macrophages that, when infected with coronavirus, become over-inflammatory, start to produce a lot of cytokines, lose their M2 polarization, they decrease the expression of lysozyme or of tetrahydrobiopterin synthetase, and by means of this contribute to the inflammatory condition. So by means of this, the lessons learned from the fatty tissues showed that the fatty tissues actually may perpetuate the virus infection, being a minor but productive reservoir, but particularly drew our attention towards the importance of these inflammatory resident macrophages that are quite an abortive reservoir of coronavirus. So the coronavirus does not replicate in these cells, but when the cells become infected, they turn out hyperreactive, producing a lot of cytokines, contributing to the cytokine storm linked to these severe disorders. So I would like to conclude also a little bit addressing the study, what has been said by the previous speakers. Pathology provided a lot of insights about COVID-19. By application of this simple technique of examining the human body, systematically collecting tissues and applying also cutting-edge technology towards those collected tissues, we were able to contribute to our understanding of this disease. So the disease is biphasic or even triphasic, indeed an aerogenic disease that targets the pulmonary vasculature, but also showing a systemic spread. It starts with the virus infection, replication, and spread. Goes over an interferon response with linked coagulopathy, complement activation, and immunopathology, and paves the way towards multiple sequela that we're now recognizing as long COVID. Many of the observations here provided useful basics to develop specific phase-specific therapeutic targets. The changes in the lymph node suggest that COVID-19 is more than just vasculocentric disease. It's a very immunoderegulatory disease. Obviously, the human body is not that designed, evolutionary designed, to deal with single-strand positive RNA viruses like this one. The B-cell memory is perturbed. There is morphologic evidence and clinical evidence of significant macrophage activation, but most importantly, of a significant T-cell deregulation that may pave the way towards reactivation of latent virus infections, especially Epstein-Barr virus, and pave the way towards developing of autoimmunity. Nevertheless, some potential targets for pharmacological interference have been detected, and they may help us to wisely choose adjuvants for long-term immunologic memory. Addressing organs older than the usual ones may also provide useful information to understand why diabetes, for example, increased in COVID-19, why obese people suffer from COVID-19. By means of this, all what I have reported here wouldn't be able without the help of many of you. I would like to mention Yasmin and Thomas, who not only authored many of the papers that we published, but stood with me in the autopsy theater to collect these tissues and perform the autopsies. Matthias Matter and Kirsten Merz provided enormous ideas, modern techniques. Matthias was the one who actually wrote the grant, and without him, we will never become granted by the BRCCH, because I didn't have time at that time by being involved in the autopsies. Jan Schneeberger performed a lot of technical work. Without the organizational support of Anna Stalder, no international collaboration would be ever possible, and we probably would have missed to link our RT-PCR data to all other data that we collected. Karl Zinner performed the statistical analysis, and many of the nicer or more nicely illustrated studies were performed in collaboration with the Stanford group. And finally, nothing out of this would function without the generous support of our sponsor. Thank you very much, and I think I hand over.